And welcome back to the second part of your cell division PowerPoint. Before we get into mitosis and cytokinesis here, there are some important cell structures you need to know about that are necessary for mitosis and cell division to occur. So we're just going to go through a few things here. First thing you need to know is a chromatid. A chromatid is an individual strand of a duplicated chromosome. One of the things that is going to happen, or one of the things I showed you in the previous things, is that, or told you, chromosomes like to pair up, and during replication, they copy themselves and they make double-stranded chromosomes. Each individual strand of a chromosome is known as a chromatid. So you'll need to remember that. The centromere is the place where those two chromatids combine. Okay, and I had showed you that and mentioned that in the previous video also. And finally, centrioles, or not finally, but centrioles, these are the tiny structures that develop in animal cells that help with cell division. They play a very important part. You have spindle fibers that will attach themselves to the chromosomes and the centrioles, and the centrioles will pull on those spindle fibers, separating the chromosomes. And then, as I mentioned, you have a spindle. That is a fan-like microtubule structure found in animal cells. And again, these fibers attach to the centrioles and chromosomes to help pull the genetic material apart or split those double-stranded chromosomes. Okay, so do remember this terminology. I will be talking about each as I move through mitosis here. Okay, so let's get going. Mitosis, the M phase, mitosis and cytokinesis. So, mitosis is divided up into 4 stages or 4 steps. The first of those is prophase. During each of these steps, there are certain things that have to go on to prepare the DNA and to prepare the cell to separate the DNA and get ready for cytokinesis. So what happens during prophase? If you look closely at the picture, you can see kind of the things that will be going on. Okay, it is during prophase that we can now see our chromosomes. Okay, again, the chromatin has wound itself up into individual chromosomes here. Those chromosomes have been duplicated or replicated during the S phase of interphase. So you can see that each chromosome is double-stranded. Okay, and we can visibly see those. The centrioles that were created during G2 will move to opposite sides of the cell. You can see those right here in the picture. Okay, right here is a centriole. Here's a centriole. They are moving to opposite sides. Eventually they'll end up on opposite sides. Okay, some other things that occur. Whoops, went too far there. The nucleus, the membrane here, begins to break down. That's very important, okay, because we need to get these chromosomes free so that these spindle fibers can attach to the chromosomes. And they are going to attach at this point that I mentioned earlier, where the chromatids are held together at the centromere. Okay, so actually I should have put that the nucleus breaks down first, and then the DNA strands attached to the centromere. But these are all the things that are going to happen during prophase. Once those spindle fibers have attached themselves to the chromosomes at the centromeres, we will be ready for our next phase of mitosis. And that next phase of mitosis is called metaphase. And you can look at here at the picture about what happens during during metaphase. Okay, the spindle fibers have attached themselves to the centromere, and what is going to happen is those spindle fibers are going to pull the chromosomes to the middle or the equator of the cell. So for me, it's very easy to remember what happens during metaphase. Anaphase starts with M, chromosomes go to the middle. So M for metaphase, M for middle. Again, this is the big thing. The spindle fibers are going to shorten up and on both sides, and that is going to pull the chromosomes so that they can line up in the middle. And this lining up in the middle is very important. This is what's going to allow us to separate the genetic material equally into the 2 new daughter cells. Once these chromosomes have lined themselves up, then obviously we need to split them apart, and that is what you see here in this next stage, which is called anaphase. Okay, you'll notice that the cell has begun to lengthen. Okay, it's more oblong or more oval than round. Okay, again, the centromeres are pulled apart. And the chromatids begin to separate. So we have gone from chromosomes, double-stranded, to chromatids. Okay, and the spindle fibers continue to shorten and pull these chromosomes towards the poles, okay, or towards the centrioles. Okay, so again, anaphase is simply the separation of the chromatids. Okay, eventually these chromatids will reach the opposite poles, and this is telophase. And you can see here some things that are happening in telophase. One of the big things that has to happen is the nuclei reform, the nuclear membrane reforms around the cluster of chromosomes, the chromatid. So we now will have two nuclei in one cell. Okay, two nuclei will form, each nuclei containing half of the genetic material. The chromosomes will begin to untangle and form chromatin once again. Okay, since they have been separated into two nuclei. And the spindle, since it is no longer needed, it has completed its job, will begin to break down. Also, you will see here that the outer membrane of the cell is beginning to pinch inward. This is called cleavage. Okay, so the cell membrane will begin to cleave. Okay, so again, prophase, chromosomes appear, the membrane breaks down. Metaphase, the spindle fibers attach. In metaphase, the spindle fibers pull the chromosomes, the double-stranded chromosomes, to the middle of the cell. Once at the middle cell, we go into anaphase, where the chromatids are split apart and pulled towards opposite poles, and that gets us here to telophase. The spindle fibers are going to unattach. The spindle is going to break down. Nuclei will form, or nuclear membranes will form around each cluster of chromosomes. Okay. And the cell will begin to cleave inward. Okay, again, the cell will begin to cleave. From there, we enter what is called cytokinesis. Kinesis means to divide or split, cyto for cytoplasm. So this is literally when the cell's dividing. Okay. This means that the two cells will be identical to each other, each containing an equal number of chromosomes and identical chromosomes. Okay, so we get two identical cells. This is different in animal cells and plant cells, and you should remember why. And I know I talked about it in the first semester. I'm sure Mr. Hess did the same thing. Okay, but if you remember, plant cells have cell walls. Okay, this is why they don't need the centrioles. Okay, but remember, animal cells do not have cell walls. Okay, but as far as the genetic material and separation of the genetic material, everything is pretty much the same. Plant and animal cells both go through the 4 steps of mitosis the same way. The big difference is in plant cells, there is going to be the formation of that cell wall. So you get in this center point here something called a cell plate. Okay. So you have the formation of a cell plate, and that is cytokinesis. Okay, so quick overview. The stages of the cell cycle. We have the interphase. That is during interphase that we have G1, which is generally for growth. We have the S phase, that is when the DNA replicates itself, and then we have G2, where any errors are fixed from replication, and then the cell prepares itself to divide by creating the necessary organelles that are going to be used, such as the spindle. And once all of that is done, we enter the M phase, consisting of prophase, metaphase, anaphase, telophase, and cytokinesis. As I mentioned in prophase, the chromosomes become visible, the nuclear membrane breaks down, the spindle fibers attach themselves to the chromosome. Okay, that will lead us here then on into metaphase, where the spindle fibers shorten and pull all the chromosomes to the middle of the cell. And then as we move to anaphase, the spindle fibers continue to shorten, and this separates the chromosomes into chromatids, and they move towards opposite ends of the poles. And once we get to the opposite end of the poles, we enter telophase. Nuclear membranes form around the clusters of chromosomes. The spindle breaks down since it's no longer needed, and the cell begins to cleave. And finally, cytokinesis is the division of the one cell into two identical cells containing all the same number and type of chromosomes. Okay, from here you will need to go again to page 279 in your textbook, and you will need to complete the worksheet that you can download from the module. So get that completed and have a great day.